The purpose of this lab is to determine the unknown salt concentration and dye concentration in the brine solution. This is done without directly testing the brine solution and done through a material balance around the mix point.
Yeah.
We started doing a serial dilution using the dye to make a calibration curve. We did 9 milliliters of water with 1 milliliter of dye.
Next, we put our serial dilutions into cuvettes and then put them in the spectrometer. We did this because we need to create a calibration curve, and we used this spectrometer because we did not get good data from the other one. Next, we did a serial dilution of the salt solution, starting with 90 grams of water and 10 grams of salt.
Next, we transferred the salt solution into a beaker so that we could measure the conductivity. We did the same thing with each of the dilutions so that we could make a calibration curve.
So to start off, we have to mix the solution in order to make sure that our salt concentration is constant throughout the solution. Then we have our peristaltic pump set to 50% speed, which we've tested and verified the flow rate for. We also have our rotometer set to 2.8, which according to the calibration curve gives us another flow rate. They both are mixed here and come out the end of this hose. We then collect our solution into this Erlenmeyer flask. So in order to turn on the system, we set our rotometer to run at constant speed. We allow the peristaltic pump to come to steady state after a couple of seconds. We then collect our sample for approximately 30 seconds. We then stop collecting our sample and turn off the peristaltic pump in order to preserve the solution. At that point, we can then use our sample in order to test our conductivity and take a small sample, put it in a cuvette, and test it for absorbance.
After thorough experimentation, we determined the solution to be 6.1% salt concentration by weight and 0.065 concentration of dye by volume. We determined this to be correct because after 2 horribly failed experimental tries, Jeff finally told us that our numbers were correct for the 3rd.
But in reality, we know that our answers are correct because after a couple of tries, we got good practice making calibration curves and using tap water to make both calibration curves for salt concentrations and dye concentrations. We were able to get reasonable results that Jeff did confirm for us to be correct.